Good morning, AP Bio. All right, it's day 70. This would normally be right before our Eco Blitz would start, but this time it's right before the AP exam. Yippee! Our target today is to go after the sliding filament theory and explain how actin, myosin, troponin, and calcium ions interact in muscle contraction because it's super complex and cool. Now, key things that we should notice, of course, for this week, you got some GR50 to make sure you got done. It's that sensory one. It's probably one of the most fascinating GRs there are. And then the 2018, which we'll be looking at on the 12th. Hope to see you there at the review. Gearing up for the test. Now, these little twerps, if you've ever tried to take a picture of three little boys under the age of, I think they were under the age of 3 at that point, you know it's really hard to get them to sit down. Right there, they've got a lot of energy built inside of them. They have the potential for action. So as soon as you get your picture, if you're lucky, they will fire upwards and then run away in different directions because they are loaded with the potential for action. And that's why an action potential is called an action potential. It has the potential to cause something to happen, for action to happen. And if that makes you nervous or gets your nerves firing, that's what today's first part is all about. And the other part's about how the muscles react to that. Cool. So our target today, of course, is first to recognize that when we look at nerves and when we look at things like membranes, there really is no negative that we have to worry about. There's positive and negative sides to, say, a membrane, but really what we're looking at is a voltage difference. So it's about more positives, that's the positive part, to less positives, that's the negative part, but it's just positives, right? There's a surplus and a lower amount, right? So always think about that, and of course things always want to go from where there's more to where there's less of them, right? From a positive towards a negative. That's how voltage works in batteries as well, only with negative things. Cool. So remember, more positive, less positive, it's about the number of ions on the side of that membrane. Now, I'm going to get rid of my big ugly head, and we're going to recognize that you should be able to draw and explain, if you've gone through that GR, the steps of an action potential and the graph that goes along with it. So right now you're going to pause me. You're going to take a stab at it on your lonesome. You should definitely give yourself a large chunk of space to do this, as always, right? Because Neil loves to talk. And then we'll tackle it right now together. Now, if we look at this, of course, one thing I should recognize is that I have a graph, and that graph is going to measure some things. One of the things it's going to measure is basically the graph is measuring the charge inside of the nerve cell, the neuron. That's crucial to recognize. And charge, of course, can be either negative, which is just less positive, like negative 70 is a pretty important number. Negative 50 is a pretty important number. Or it can be up in positive land, like positive 50 are important numbers, right? And then this would look at what's going on the nerve over time. Behavior over time graph, right? So if I look at a cell, a nerve cell, and it's ready to go, it's got potential, it's got this thing called resting potential. And being at resting potential, that means that when I look at the cell, this is the only part I care about right now. This is the axon, right? South of the axon hillock. And if we look at this, if this cell is ready to do work, it is, well, positive on the outside, more positive on the outside, and less positive on the inside. We say that it's at its resting potential. We'd also notice that it has two different poles, two different sides. So right now, this cell is polarized, right? It's kind of like a hydroelectric dam. It's ready to flow, right? So this should definitely make you think, damn, as in hydroelectric dam, things flowing down their concentration gradient. Yeah, I just said that. Now, key thing, why is it that way? It's because the cell's been getting itself ready to make itself a salty banana. It's put salt, sodium, on the outside, and a different type of salt, banana salt, potassium on the inside. So if you always remember, it's salty bananas, that's more salt, sodium on the outside, and more potassium, like from bananas on the inside. Not mine, but a very handy thing, right? And the way that is because there's 3 sodiums for every 2 potassiums if things are looking good. But that's why, creatures crave salt. They need it to fire their nerve. Now, if a cell looks like this, a neuron looks like this, then it means the inside of the cell where the sensor is reading will be down in negative land. It'll be in the less positive land. That's step 1 right here. This is the resting potential. This is a nerve that, like those little boys, has the potential for action. It's fully loaded up, right? Then what happens? Well, you see a bear. Something cool happens. Or if you're like those kids, squirrel, and you run for it. Key thing is your sensory systems pick up on some kind of stimulus, and if it's big enough, that'll cause enough to build up to activate the nerve. And the first thing that happens with that, of course, is these things called sodium channels open. Now, if they open, that's a crucial thing. Here's my nerve. It was all set up and it was ready. It had positives on the outside, sodiums, and negatives on the outside. But key thing is one of those channels open, and that allows the sodium to rush in. So if sodium rushes in, because it's just going down its concentration gradient, that's going to bring more positive things to the inside. That means it's going to rush its way in and make the inside more positive and the outside less positive. Hey, it's just diffusing. It's simply diffusing by passive transport. But that means that this part of the cell right here has a different charge than the rest of it. It was polarized. Now it's starting to depolarize. And it's called depolarizing or depolarization, right? Now, the sensor, of course, is just picking up on the nerve, and we can see that only part of the nerve's axon has actually become a little bit more positive, and that's why it creeps up towards positive land. And we see this little bump right here. That's what's going on as the first sodium channels actually open. Now, crucially, that is reaching very close to this point called the threshold. And if enough sodium channels open up to rush in some of that, the very first sodium channels, then it can reach up to negative 50 millivolts. And that's what these are. It's the millivoltage of the inside of the nerve. If that happens, then that charge causes the other protein-based channels that are further downstream to change shape, because charge makes proteins change their shape. And that's what allows a crucial thing. If they change shape, that means that more sodium channels can now open. And that's crucial, because if more sodium channels can open, more sodium can now rush in. Going down this concentration gradient, we now have what's called a rising action potential. And that means that the nerve has reached the threshold where it can actually fire, right? Now, at this point, we've got lots of sodium channels that have opened up, and sodium ions are rushing to the inside of the cell to make it more positive and the outside less positive. It's truly depolarizing. We now have an action potential. That's going to cause voltage changes downstream of it, this way on the nerve, which is going to cause the action potential to move like a wave down that nerve. Now, crucial thing, down the nerve, it's resting potential. It's resting and ready to do this action because it's still got more sodium out and potassium on the inside of it, right? So I can see this happens on the graph because when I'm measuring that, now I'm measuring the inside and it's gone from negative land, less positive, up to positive land because I got sodium rushing in. And that's the third step. That's the action potential. That's the fancy word for the nerve is firing. The potential has now become action. As it becomes that action, and I take a drink of water. One cool thing about this is now my nerve's firing. It's doing the wave down the line. What else has to happen? Well, I got a bunch of sodiums that are rushing in. That changes the charge. And when you change the charge around a protein, it changes its shape, its conformation. Now, that charge change is now so positive that it actually closes sodium channels, which makes sense. They did their work. We want them to shut, right? And it also crucially now opens potassium channels. And that allows potassium to do something it hasn't been able to do yet. It allows it to go out. It lets the bananas out, right? This is what's known as the falling phase. And in the falling phase, that's crucial. The action potential has migrated its way quickly down the nerve. It's super fast. And that action potential has allowed some crucial things. The sodium has rushed in, allowing that to fire down. It's going to go on to the next nerve, send off some neurotransmitters, do its work for you, right? At the same time, you've got a problem because now it's not a salty banana. There's a banana with salt there. And that's because now the potassium is moving out. And that makes it, well, more positive on the outside and less positive on the inside. Again, hey, it's repolarizing. It's kind of like the cell is trying to reset itself in some fashion, right? Now, you can see that on the graph because when you're measuring it and you're measuring it here, that's going to make it go back down to negative land. And actually, it does it so well that it pumps out so much or lets out so much potassium that it actually becomes more negative on the inside, less positive than it was before, and creates this thing called the undershoot. Because you let out more potassium than you would think you'd need. But that's a good thing because that's getting you ready for the next part. Now, the problem is I now have a nerve that's fired, but now it's a banana with salt in it. And I don't want that. I want a salty banana. And that's why, of course, we need this other thing that is not, of course, a channel. It is a pump. And that is called the sodium-potassium pump. And its job is beautiful. It is to reset it, right? Basically, what it's going to do is it's going to pump it. It's going to pump 3 sodiums out for every 2 potassiums it can pump back in. And that's why you let extra potassiums out, so you can always get the salt, the sodium back out. And eventually, of course, if you put enough ATP into it, because this is active transport, you're pumping them against their concentration gradients, you'll end up with 3 sodiums for every 2 potassiums. Hey, look, it's back to being a salty banana. It's back to the potential for action. It's at its resting potential again. And you can see that on the graph, because as it does it, measuring the inside of the cell, it comes back up a little bit, adjusting the ratios. So now that the sodium and potassium are in a balance of 3-2, a little bit less positive on the inside. Potassiums are on the inside. The pump has just reset this thing, and that's level 5 of this thing, right? Cool. Your nerve is now ready. It's now a reloaded hydroelectric dam. It's ready to fall downhill and fire nerves. Now, key thing we should notice, of course, to do that, it's going to take ATP. This is why you were exhausted after a hard day of learning, or I'm tired after a day of teaching, because all that neuron reloading and reloading of the hydroelectric dam, that's where most of our ATP goes. And that's why nerve cells are so expensive, just to pump sodium out and potassium back in so you can let it fall back downhill. Kind of makes sense why you're so tired. Go get a candy bar or something. So hopefully you're recognizing this should make you say damn, as in hydroelectric dams, right? And with that, it should also remind you of dominoes, or my favorite analogy, this is like the baseball, or originally the soccer wave. And this is the idea, of course, that you're sitting there and in a stand, you paid way too much for a hot dog, and suddenly somebody next to you stands up. Well, they're like the sodium moving in, right? And then the next person next to you, by peer pressure, like, oh, I should stand up next. And soon you've got this beautiful wave that will carry all the way around. But you don't do it once, because after you stand up, you sit back down. That's like the potassium moving back out. And then you eat a bunch of salty nachos so that your nerves and muscles can now do the next part. And that wave is the action potential all the way around, and it can be cyclical. So think of analogies that are pretty handy for your brain. Now, other parts we should recognize, that's exactly what's going on here. I've got a bunch of sodium and potassium channels. They're going to rush to get up, and it's going to fire off like a nerve all the way down the wave. And it takes a lot of ATP, energy, to reset them. That's why you should thank your parents every day, critters. Now, I'm going to show you this video because it was put on by Pearson, and they don't know I'm doing it, so hopefully I won't get in trouble for copyright. Yeah, right. Now, key things about this, of course, as we're watching this thing, think about that Mexican wave. That's where it started in soccer.
This baseball player springs into action, his nervous system rapidly processing information in a network of nerve cells, or neurons.
Now, what is this? This, of course, are synaptic connections. These are memories. This is where I spend all of my life trying to get your brain to scar up and have these beautiful scar connections between them. So nerves can talk to other nerves, and the more nerves that talk to each one, the smarter you get, and the easier it is to recall memories. Hence all the annoying jokes.
Each neuron receives input from one or more cells. In response, the neuron may generate an electrical signal known as an action potential that travels down the length of the axon. Let's take a closer look at the plasma membrane of an axon. Even without an action potential, the axon is a busy place with many ions moving across its membrane. Much of this ion movement is driven by the sodium-potassium pump.
Hey, look, it's
using energy from ATP, sodium-potassium pumps actively transport sodium ions out of the cell and potassium ions in, creating an uneven distribution of charge across the membrane. Some potassium channels are open all the time, allowing potassium ions to leave the cell. As a result of these ion movements, the inside of the cell is negative relative to the outside. This condition is called the resting potential.
Hey, look, salty banana. More sodium outside, less positives inside. There's the banana, right?
The membrane of an axon is also packed with gated ion channels that open and close during an action potential. At resting potential, the gated channels are closed. If a stimulus changes the distribution of charge across the membrane sufficiently, the gated sodium channels open. Movement of sodium ions across the membrane makes the inside of the cell more positive.
It's depolarizing. Hey, we got a stimulus. Now that depolarizing is going to change charges, which makes proteins change shape.
This reversal of the charge distribution causes the gated sodium channels to close and the gated potassium channels to open. As potassium ions move out of the cell, the original charge difference is re-established across the membrane, closing the gated potassium channels. This sequence of events is called an action potential. The sodium-potassium pump restores the distribution of ions back to their previous levels at resting potential.
Here comes the awesome part.
Let's look at how the action potential is conducted along the axon. As the change in charge difference across the membrane spreads from open sodium channels, other sodium channels farther along the axon begin to open. The original sodium channels close, and adjacent potassium channels open.
And look, it literally is a voltage wave, where the charge potential over the membrane is changing and doing the wave from one domino to the next, from one sodium channel to the next, and resetting with the potassium.
As potassium ions move out of the cell, the original charge difference across the membrane is restored, and then the potassium channels close. Meanwhile, new sodium channels open, followed by the opening of new potassium channels and the closing of sodium channels. In this manner, the action potential is propagated along the axon of the neuron, eventually reaching another cell. The information carried by this action potential will be processed with other information, permitting this baseball player to make a spectacular catch.
Unless he's a Mariner. Oh, bad joke. Now, cool things about this. You should be getting out GR48. The second part of that is our focus today. So pause it, grab it. If we look at this, this is very much about putting the AP in action potential, right? Because it's nerves that take you to do well in test, and the idea of how learning works. Now, key parts about this, learning is all about synaptic connections. Why have we been trying to scar you all day? So we can make one nerve touch many other nerves and increase the number of synaptic connections you can make and how fast you can retrieve those. Do not forget that nerves, of course, when they're ready to do work, they are salty bananas. More sodium's outside, potassium's inside. Fortunately, our diet supplies us with enough of that, too much of it, that we don't have a problem with it. But things like mountain goats are always looking for salt licks, because they need to make sure they have salt and potassium, sodium and potassium, to fire their nerves and muscles. Now, you can see, of course, in the video, it shows this beautiful sodium's in, carrying down, open up the next channel, goes like a wave down as it depolarizes. Then the potassiums move out as it opens up those channels to repolarize it, and then it resets using the sodium-potassium pump. You should make sure you understand this graph very well about how it's measuring the inside of the cell to go from one sodium channel to the next, resetting with the potassiums, and then totally resetting with the sodium-potassium pump. Now, learning and nerve excitation. Key thing about nerves is they are hopefully connected to multiple nerves. That's why they look like trees, the dendrites that are connected to synapses. As we have those axons come down, this is kind of like with a TV. If you watch TV too much in the last couple of weeks, like people always do, one thing we can notice is you don't really pay attention to the commercials. And if you listen to English class too many times, you don't really pay attention to that. That's because those aren't creating enough of an actual charge difference to get your nerve excited, so you just ignore them. It's kind of like my voice right now. On the other end of that, let's say that we have a commercial where you keep seeing it over and over again, like McDonald's commercials, because they use SCAR all the time. What they're doing, of course, is they're repetitively showing you that creepy clown, so that's enough messages that your brain's like, oh, I should pay attention to that, and that fires the action potential. So the more times you are exposed to something, that's like the repeat part of SCAR, the R, that would cause you to have a better chance of your nerves firing and your actual memory working. Now, SCAR exploits this because, of course, you don't have to have one signal that sends it. You could connect two signals like they've done, like you drive down the street in a giant truck with a huge greasy hamburger, or you smell the beautiful smell of French fries, and you think of that clown that creeped you out when you were little. All those different types of connections, neural connections, they allow them to access your brain and sell you more of those wonderful Happy Meals that are so delicious because they actually fire your nerves. Advertising is all about SCAR, so we've been exploiting that. Now, key things about this, of course, it also can work against you because other nerves can cause inhibition. If you're learning salty bananas and you're like really scared of your AP exam, that could fire other nerve impulses that prevent you from making your memories. So relax, people. It actually helps you learn, right? Now, key things, that's called inhibition, which kind of makes you think of inducers and things like preventing stuff. Heterozygotes, just allele uneven. One of the best ways, of course, to make memories is to tickle this thing called the amygdala. That's the emotional part of your brain. That's why I've made so many cheesy jokes this year or scared you to death with quizzes because, things like fear and things like humor can trigger more synapses to form because that helped their survival in the past. That berry's good. That person made me giggle. Maybe I'll mate with them. Or, oh no, that bear's going to eat my kid. Oh, all those things help memories. Now, ways that this can happen faster is nerves inside of your central nervous system can develop a fatty layer. That's the white layer versus the gray matter. Gray matter, of course, is what Einstein's famous with. Those are nerves that are slower, but you can pack them more densely and make more memories. They're in your brain. If we go to things like your spinal cord or your peripheral nervous system, we find a lot more white matter, or the nerves that are connected to them. They basically have insulation that allows them to fire faster because you got to send a message from your brain all the way to your toe, right? Now, there's oligodendrocytes, the straight ones that you find in the central nervous system, and then there's Schwann cells that you find in the peripheral. Things like squid do the total opposite. What they do is they have big fat nerves with no fat on them. They're like big wide ones, and that increases the surface area so they can send their signals faster without the fat layer, but it allows them to actually have a greater amount of ions moving. So it's a different technique than us, and invertebrates found their own way to do it, but they have their own intelligence, right? They can change their skin with it. We tend to use these myelin sheets to insulate our wires, basically, and it sends a faster signal. That's why fat needs to be part of your diet, and why probably opening up fat to our diet made our species faster and smarter. Now, this is basically works in connection with these things called the nodes of Ranvier. And if we look at those nodes of Ranvier, what they do is they basically allow the cell to have leapfrog go on. The sodium channels open upstream of it, but then they can't make their way back out because the fat prevents the charged particles. It's still about hydrophilic and hydrophobic, so they diffuse faster through the cell to the next node, and basically they can skip from one node to the next and fire the nerve faster, so your reflexes can kick off quickly, or you can, wiggle your finger really fast, right? Awesome. Get fat in your diet. It's good for you. Now, typically what we do at this point is we do a check for understanding quiz. Flip to a new page in your ETD. Let's see what you know, right? If you did GR50, then you should actually know what actin, myosin, tropomyosin, troponin, calcium ions, and myosin binding sites and ATP all have in common. I know you don't have a bio, buddy, but you could always pause me. This is usually about the point where fear kicks in with kids, which is useful for learning too. And then we help each other out. All of these play a role in muscle contraction. So get out GR50, right? With muscle contraction, big things we should notice, this also has to do with nerves talking to them. So this is about stimulus, integration, response, because 50 is about sensory and motor reactions, right? It's always stimulus, maybe some integration unless you got a reflex, and then response, which we've hit before. This is about how energy waves in the universe, like sound waves, heat waves, light waves, are picked up by our sensory nerves. They, of course, are afferent. They're attacking us so we can figure out what's going on. Our brain integrates those, causing a bunch of action potentials inside of our nerves. So we go from electromagnetic waves or sound waves to, electrochemical waves, action potentials, which then leads to our behavior, our response. You flex a muscle, for instance, right? You pee your pants. Now, how do muscles contract is the other big part of GR50. It's ultra complex, and that's why the AP loves it. Plus, it's really fascinating to understand. Now, muscles do work, because they, of course, can apply a force over a distance. That's the definition of work. And muscles basically do two things. They can either contract and get shorter or relax and get further, right? They can flex or extend. If you're an invertebrate, then your skeleton's on the outside giving you lever arms. If you're a vertebrate, then it's an endoskeleton. It's still there to give you, of course, something to do your work on, and gives a mechanical advantage, right? Now, muscles are super complex and good in your diet because they are loaded with many, many cells with lots of protein in them. Now, a muscle, of course, is made of smaller units that are called muscle fibers. Each muscle fiber itself is made of single muscle fibers, which are muscle cells, myocytes. They have a nucleus like any other cell, but the cool thing inside of them is they have these little bungee cord things called myofibrils. Myo just means muscle, right? Inside the myofibrils, there are singular units called a sarcomere. Sarcomere means flesh, so they're in the flesh muscles, right? And they contain these other really cool things called actin and myosin. Now, I always remember that actin is acted upon, and myosin is the mover. So, scar your brain and help it out. Now, muscles are made of the actin and myosin because you need the interaction of these little motor proteins, those clown feet from the Harvard video I showed you a long time ago, that are going to interact and move on top of basically a tug of war rope. which is the actin. It's acted upon, and then the little creepy spider feet, they move on it. That's the myosin motor heads, right? There's also different sections of the muscle, the Z band and the M band. And basically what you have here, I'd normally do it in class where we show a tug of war. The Z band, of course, is just attached to the rope. The motor heads, the myosins, are right here. And what they do is they crawl along it and they cross and get shorter. So the whole muscle unit, the two Z bands come together, it becomes more dense, which is why it looks darker. And you just contracted it. Relax those feet and let them slide back, and it will slide back its other way. It's called the sliding filament theory of how muscles contract. It's a pretty good theory. I would normally show this picture, but I will put the link on. You should watch it. It shows the tug of war and describes it quite well with the creepy spider feet called the myosin heads. Now, how do muscles know when to contract? Well, that's easy. You got a stimulus, you got integration, and you got response. It's all, of course, connected by neurons. And our neurons, like motor neurons, come out from our brain. They send an action potential, which sends neurotransmitters across to the cell. And then your muscles themselves have their own action potentials. Even plant cells have action potentials, which is pretty cool. If you got a membrane, you can build up a voltage difference. That will connect with a thing called the sarcoplastic reticulum. Man, yeah, tubes with muscle flesh. And then calcium ions. So if we look deeper into this, the nerve has fired enough to make these synaptic vesicles of acetylcholine, a major neurotransmitter in your body, makes you feel good as well as makes your muscles move. They go across ligand-gated ion channels. That lets in things like sodium ions, which then cause an action potential in the surface of the muscle fiber itself. That goes down a thing called a T-tubule. Hey, it's shaped like a T, which brings it down into the cell and connects it to the sarcoplasmic reticulum. It's like the endoplasmic reticulum, but it's in the muscle, the flesh part of it. And it's basically a big storage bag for lots of calcium ions. When this charge comes down, it opens up a bunch of voltage-gated ion channels and lets calcium go from high to low. It's like it's all connected this year. That will then float in and interact with where the actin and myosin are down here, controlled by things like tropomyosin and troponin. Now, cool thing about this, tropomyosin basically acts like a giant rubber band that sits over the surface of the actin at its myosin binding sites. It's in the way. It's an inhibitor. It's controlled by a control switch called troponin. As calcium floods in, it has the right shape and charge. It binds with troponin and turns it off from being onin. That basically causes and allows for that calcium to bind to it. It shifts the tropomyosin out of the way, and that exposes and opens up the binding sites so that myosin motor protein heads can attach to them and start to do work. That's the easy first part of this. Now, I would regularly show you this video. I'm going to put the link on. You should watch it. It's one of the best ones for explaining how muscles work. It shows all the parts. And it's not me narrating, which is a nice break. Now, what I want you to do right now is I want you to go and get a rubber band and put it on your wrist. Now, this would be much better if you could see me, but we can't bring back the video. Key things with this, of course. Pause me now. Go get a rubber band. Put it on your wrist. This is the model I came up with in 1997 when I took the exam, and I still remember it, so it must work, right? Now, if you take that rubber band, you put it on your arm is the actin. It is acted upon. The rubber band is tropomyosin. It's in the way, and it's going to go right over your wrist, which is the myosin binding site. In addition to that, you got your other hand. The pointer finger on your other hand is the calcium ion. And, of course, the thumb is the myosin motor protein. So the first thing that happens is this is in the way, right? The tropomyosin is blocked, troponin is turned off. You get a nerve that fires. Calcium ions are released from the sarcoplasmic reticulum, and your pointer finger would touch and bring calcium to this cool thing called the tropomyosin, or the troponin, and that would cause tropomyosin, the rubber band, to slide out of the way. Now your wrist is exposed, exposing the myosin binding sites. Now there's another thing that has to happen here. One of the things that's happened is you got to get some orange slices from mommy at halftime so that ATP can flood in here and bind with the myosin motor protein head. Now the ATP binding causes the ATP to change the shape of the myosin head, and it cocks it back as it's turned into ADP and P. You just use energy to cause change in your body, chemical energy, right? The ADP and P are still bound to this, which then causes it to have the right shape to bind with the myosin binding site. When it connects with that, that causes the inorganic phosphate, which should have an I because it's not connected to anything anymore, to pop off into solution, and that causes what's called the power stroke. As it does that power stroke, that kicks out the ADP and slides the whole myosin crawling up your hand. Now, if that was all that would happen, it would be stuck there. But the good news is if you've got more ATP from the orange slices, that ATP can now come in, bind to it, and recock it and disconnect it. Now, if you've got a bunch of those doing it, it will crawl along like a creepy little set of spiders all the way up it and make the whole actin, the rope, get pulled further down. Now, key thing is that would allow you to go through all the cycles. So practice that a few times on your own. It's a lot easier when you can see me, but hey, coronavirus, thank you very much. If you think about it, this is a lovely thing you can use anywhere, even when you go to med school, but even during an AP exam, because, you know, you got your thumb in your arm most of the time, right? So go back to your quiz. See if you can explain the function of each of these. Pause me and see if you can do it. Use your model. Don't be afraid. Cool. If we look at this and tackle it together, we can see some key things. There's your arm. There's the rubber band. There, of course, is your pointer finger. And here comes your thumb, little ATP. If you look at this, the actin is acted upon. It's the rope. Myosin is the mover. It's the motor protein. It's your thumb. Tropomyosin is the rubber band that's covering up the binding sites, so it can't do any work on it. It's controlled by troponin, the on switch, right, which can be triggered by calcium ions, the inducer. It's basically the ignition button. It hits that thing and slides it out of the way, and that exposes the myosin binding sites, which would be your wrist, so they can now bind with, of course, the myosin head, and ATP would be the thing allowing it to, do its power stroke, turning from ATP into ADP. PI gets kicked out, ADP goes with it as it power strokes, and then it kicks out ADP, opening up a parking spot for the next ATP, unless, of course, you didn't have the orange slices. Thank you, Mommy. Now, why doesn't anyone like sore muscles? Because they're so sarcastic, just like me. Now, key things about this, of course, people tend to ask, okay, why do I get cramps? Why do I have to stretch before I go and exercise? Stretching is because, of course, if you don't get enough blood flow into your muscles, then those myosin binding sites can be all parked up with the myosin motor heads. That can cause your muscle to basically be stuck together. When you're stretching those two things apart. In addition to that, of course, why would I want to make sure that you get lots of electrolytes? I'm drinking my Gatorade. That's because that helps to balance out the ions. If I'm dehydrated, then, of course, the calcium ions can't flow as well. The action potentials can't build up in the muscles, and then you would, of course, not be able to flex them. Hence the cramping as they get stuck together in a stuck tug of war. And if you don't get enough ATP because you don't have enough blood flow because you haven't been exercising enough, then you're not going to be able to disconnect them and they're stuck together. It's almost like your body makes sense, right? Now, typically at this point, we would go into the Eco Blitz. The good news is we already did that ahead of time. So you know the story of this poor little fish and why it's so hard for him to breathe. Oh, he's going to suffocate. Now, cool thing is usually we would collect at this point ETDs 61 through 75. That, of course, we're not going to do at this time, but you should make sure you go through and complete your scars. They are your number one study guide. I just had a student write back from college and they thanked me again for annoying them with scars because they actually remember stuff and can use it. It's like they appreciate it as they mature. Your target, of course, is to get yourself ready for the exam. I'll have one more video this week. GR50 gets into those muscles. It gets into great things like the nerves. It was a lot of work and it's a lot of fun to learn that stuff. There's crickets with ears in their legs. So weird. We'll go over the 2018 FRQ, and then you'll knock the AP exam out of the park because it's easier than taking any one of my tests this year, and we've been preparing for a regular test. Oh, and this is an easy one, so don't stress yourself out. It's going to be fine as long as you've been doing your part. All right, folks, I'll see you in the next one.